We're going to do a really quick switch right now, and I'm going to introduce Dr. Tiffany Nelson, who's a research scientist who works with the microbiome, which we've been talking about quite a bit over the last 2 days. She's worked with cactus soils, leopard seals, and the human vagina.
Yep.
And her fun fact for this evening is that seal poo smells like fish. There you go.
Okay.
While this happens, I have been vested
great.
with the task of telling vagina stories, which it turns out I don't have that many. But when I was talking to an older woman, which kind of speaks to this moment, she said,
Sure,
oh my God,
yeah.
I'm going to this vagina talk. I don't see her here. Because I was in Berkeley in the 1960s when we were all taught to get out our mirrors and learn how to examine ourselves. And when I told a much younger woman this story, tonight, she sort of looked at me in a peculiar way, and I said, it was our bodies, ourselves. Yes, thank you. There was a time. So the vagina conversation's been around a while, and I'll throw to Tiffany now, and she can tell us better vagina stories.
That's putting her on the spot, isn't it? Come up with a vagina story to fill the time. So thanks, Laura. So I wanted to tell you all about the microbiome. So I wanted to tell you you're never alone. Even if you thought you were alone, you always have your microbial friends with you. In your pits, in your nose, up your butt, they are everywhere. So they are incredibly small. They're smaller than cells in our human body. They're about 10 times smaller. And so what that happens is they colonise us everywhere, and they outnumber our own body cells by a factor of 10. So they're incredibly numerous in and on our bodies. And in the last 20 years, computational and instrument technology has really advanced. It's advanced so much that the cost has come down, that everyone's just sort of sequencing every bit of microbe they can find in every sort of orifice. So we've kind of charted the human microbiome, and we now know that different parts of the body have their own distinct types of microbial communities. And these communities, we see, have strongly evolved with us over time. And the reason we know that is because when we compare, say, the gut microbiome of humans to the gut microbiome of a gorilla, we see species that are very similar. So it's not because gorillas and humans are hanging out together, it's because we have evolved with these bacteria in our guts, and they are there because they're performing a very important service that makes us greater than ourselves. We are sort of like a macroorganism. So colonization happens during birth. So usually you come out the vaginal canal and you're completely colonised by a suite of organisms. Although at the moment that's sort of being debated, because there's now an understanding that we might actually be colonised during our time in the womb. So we're learning more and more about this all the time. Over the time of our lives, the influences and the behaviours we exhibit, they really change the structure of our microbiome. So the geography of where we live, the environment where we live in, our ethnic background, these all shape our microbiome. The mode of birth that we had, whether we were caesarean or vaginal, whether we're breastfed or bottle-fed, the type of diet we eat, our exercise regime, whether we're exposed to stress, different medications, and particularly antibiotics. Antibiotics have been shown that in some cases they can reduce some of the bacteria in our gut microbiome for up to 10 years. So obviously the way that we live our lives has an impact on some of the creatures that organisms that live in our body. One thing that we're really discovering more and more is that there's an association of the microbiome with ourselves in disease. And so it's not clear yet whether this is correlative or causative in all of those cases. So whether it's just an association because you have a disease and your microbiome is changing, or your microbiome is actually changing and causing a disease. So the way we study that is we put babies in buckets. No, we don't do that, because this is just some weird photo I found on the internet, which who knows what they were doing to these poor babies. One of the ways that you can actually test correlation is looking at germ-free mice. So it's a model that is sort of been evolved to now use in relationship to the microbiome. So they live in a bubble, these mice. So they're housed completely in sterile situations. So sterile air, sterile food, sterile water, sterile bedding, everything in their environment is completely sterilised. And the way they kind of lose their microbiome is they're given a very strong treatment over time of antibiotics that allow them to have no microbes in their system. So this allows a really unique system to study the causative effect of microbiomes when we introduce them back into these germ-free models. So when we do introduce them back, for example, with mice, if we give a mice with a germ-free, no microbes in their system, a normal diet, they will not gain much weight. They will stay very lean. But if we in fact give that mouse a normal mouse's microbiome by transplanting it into the mouse gut, we will see that animal will start to gain weight with the same amount of food. It'll gain weight and muscles in the same environment. So we know that the microbiome is able to harvest energy more than our own system. So we don't work in isolation without those bugs. If we transplant an obese mouse's microbiome into the gut of a normal mouse living in these germ-free conditions, they too will they will become obese. And what we know is because that microbiome is much better at extracting energy. So given the same dietary conditions, the microbiome of those obese mice have a better ability or function to extract energy and harvest energy from that diet. The way we do this, don't do this at home, but the way we do these fecal microbial transfers is kind of what you think. You get a poo sample and you actually homogenise it, you put it in a tube and you transplant it back into the gut of the mouse. Or in some cases, it's being trialled in human conditions. So we're actually starting to look at this in humans to repair some damaged situations. So I'm going to give an example of that right now. This is a fairly old study. It's a condition. The colours represent different types of bacteria that you see in the gut. It's just abundances, that doesn't really matter. So we have a patient that's been sick for about 8 months of time, chronic diarrhoea, severe gastroenteritis, no treatment was working, severe weight loss, confined to a wheelchair. So this is a really bad condition and clinicians are starting to sort of wonder what the hell to do because this lady it's starting to look quite bad. Her partner, who also happens to be the donor of this fecal transplant, he has what we see is rather a healthy microbiome. In this case, his bacteria are much more in the range of what we consider to be normal and healthy. So he was identified as her donor to try and repair this gut microbiome in this sick lady. So this is day 0. The transplant occurred, and by day 7, we see a significant shift in the microbiome in the sick patient. By day 14 and day 33, minding that the abundances are a little bit different, you can see that the dominance of the type of groups, the orange one in this case, which is what we're interested in, and the yellow ones, are much greater. And they've completely overtaken the other types of bacterial groups. This woman fully recovered thanks to this type of fecal microbial transfer technique. But I'm going to talk about something else. I'm going to talk about the vaginal microbiome now. So the vaginal microbiome that exists in our reproductive tract. Heaps more interesting. Most of us know about it, some more than others. So you can see here is a vaginal cell, and you can see that there's a particular type of bacteria that really dominates a healthy vaginal environment, and that's called a lactobacillus. So a lactobacillus is the same type of group of bacteria that you will also find in your yogurt. It's not the same strain, but it is the same type of bug. What lactobacillus do in this environment is they feed on glycogen. So it's a simple sugar in yogurt or in milk. When you make yogurt, it's actually they're fermenting the milk sugar, which is lactose. But in this particular case, in the vaginal mucus that's excreted in the wall of your vagina, it contains a large amount of sugar called glycogen. The lactobacillus ferment this glycogen into lactic acid and other types of acid, and it's this acid that actually keeps the pH very low in that habitat. So think lemon juice, whereas the rest of the physiology of your body would be more like water. So this is rather acidic, and what this actually does is it provides a protective mechanism for other bacteria to not able to colonise this environment. So it really makes it a great, safe habitat for lactobacillus. When we look at sort of like all the sort of global microbiome, we see that most women, 70% or so, will have a dominant lactobacillus species. So usually there's just one strain of lactobacillus that will dominate, and then there's about 30% of women that suffer from what we call a low lactobacillus state. And it's these type of women that are kind of sometimes suffer other sort of symptoms that are associated with a particular disorder called bacterial vaginosis. So bacterial vaginosis is a really common reproductive disorder in globally. In Australia, it's probably about 4 million women suffer from bacterial vaginosis. It has a whole suite of negative effects, and mostly these seem to be related to this low lactobacillus dominance. So you have a low lactobacillus situation where there's less protective effects in your vaginal environment, and you're more open to colonisers. So women with BV are often more likely to get STIs transmitted. If you're pregnant, you're more likely to suffer from miscarriage, and you're also more likely to have a harder time falling pregnant. So the type of infections that you might get in a BV's case, HIV or chlamydia or gonorrhea, these are all big things that you're more likely to get when you have BV. Sorry, my voice is... I've got a dry mouth. I wanted to show you one of these sort of diagrams through time of what this environment looks like. So this is a really special contribution to science from these women. These are daily swabs collected from women over 10 weeks of life. So that's really impressive. And then in that... Thanks. Thank you. So in that environment, so again, the colours represent different types of bacterial species, and you can see in this lady, she's dominated by a lactobacillus, and we see a change or a slight shift in the types of bacteria right around menstruation. And that's not that surprising because the act of menstruation causes a huge habitat change. You know, the introduction of blood into that environment, the high iron, really shifts the lactobacillus to another dominance. But you can see that almost always they bounce back and they're constantly dominated by a lactobacillus. So this is probably a healthy situation for this person. She probably doesn't suffer too many issues. In the case of this woman, she's got another lactobacillus dominance. You can see she also has a very small shift, but it's pretty stable. And when we look at a woman with a low lactobacillus community, you can see that there's a lot of different colours, which shows that it's much more diverse in that environment. And then what we see is that at week 4, she has symptomatic BV. So she actually takes this time and goes to see the doctor, and at this case, she gets a medication. When she starts taking her treatment, you can see that it really becomes quickly dominated again by lactobacillus. So it shows that the treatment is working. When we look at this other low lactobacillus woman, you can see that the structure of the microbes are quite different. And when she also goes to get treatment, you can see that it bounces back to a lactobacillus-dominated environment. So the treatment is working, but you can see particularly on this graph that right there, the treatment's starting to stop work. And what we see in BV is that the treatment isn't very good. There's almost usually about a 9-month remission rate, and most women seem to suffer from it for very long periods of time, sometimes all throughout their life. If we kind of look at how you can keep yourself void of bacterial vaginosis and generally just having a healthy microbial vaginal tract, you want to kind of avoid some things, I guess. You don't want to be douching too much or at all. Hot baths or regular baths can sometimes be problematic. If you have multiple sexual partners and female-to-female sexual relationships, these often have higher risk factors for BV. medications and antibiotics are often more likely to see a BV case afterwards. If you wear G-strings, if you take different types of medications, and also if you smoke, you're going to be more likely to suffer from BV. So I was involved in a study that looked particularly at smoking. So we wanted to investigate a little bit further about this vaginal microbiome and the relationship it has to smoking. So we looked at 20 non-smokers and 20 smokers, and we got their extensive sexual history, and we also took 2 swabs from their vaginal tract. One was of the microbiome and one was of their metabolome. And this is like the concentration of compounds in their system, so we can find any sort of compounds that are present there. And what we found was that tobacco is the main source of cigarette smoking, or the main compound in cigarette smoking. It breaks down into nicotine, continine and hydroxycontinine. And we see that in the women that are smoking in their vaginal tract, it is much, much higher. In the women that are non-smoking, we don't see any sign of nicotine, highlighting the fact that if you smoke, you're going to have smoking chemicals that make their way to your vaginal tract. It doesn't mean that, non-smokers, passive smokers, they might be included, but in our particular case with these women, we didn't see any of these. You can also actually see other types of chemicals and indications of medications in your vaginal tract. So for one woman in the study, she'd been taking cocaine. We could see that in her vaginal tract. Analgesics, paracetamol, these sort of drugs and medications, these all show up. It's not really clear how all these different compounds can actually change the microbiome, but this is what we're trying to sort of work towards understanding. So we know that the smoking affects the vaginal tract in a compound way, or a concentration of compounds. Another compound that we found was really high were a particular type of compounds called polyamines. So these particular compounds were much higher, again, in the smokers. They still exist in the non-smokers in this particular case, but they're significantly higher in the women that smoke. And what we found is that this is also a case for symptomatic BV. So these particular compounds suffer from what we call malodor, and it's a symptom of bacterial vaginosis and also of a low lactobacillus environment. And there is actually a laboratory clinical test called the Whiff Test. It's actually a whiff, you smell the chemical and you find out, you give it a rating, and it stands the test of time because they still use it. And basically, cadaverine comes from the word cadaver, meaning dead body. Putrescine comes from the word putrefaction, meaning putrescine comes from putrefaction, meaning putrefaction. So that gives you an indication of the type of odours we're talking about. So it's not good to have these in your vaginal tract, of course. And I might add, women suffer heinously from having these sorts of problems, as you can imagine. What we discovered was that in these low lactobacillus women, so this incredibly diverse and different type of bacteria, instead of fermenting the glycogen that was readily available in the vaginal mucus, they're actually fermenting other types of amino acids. So they're able to use those amino acids and convert them into these polyamines. The polyamines are then much higher in their pH. And it's the pH that then creates a cycle of increasing the acid so that it gets closer to a neutral sort of pH scale, and this opens the door to allow other colonisers. So this could be a real mechanism to understand why these bacteria, or why women with a low lactobacillus environment are more likely to get more sexually transmitted infections. So just to recap, you are your microbiome, you always have your friends with you. There's many things that you do that impact your microbiome, but at the level of an individual, it's very hard to really understand exactly what you're doing that might impact your microbiome. And they clearly link to your health, and they're an important part. And I think the future of microbiome research is just going to show us more and more of how we can understand it and really harness it to make us feel better. And I'm going to leave you with this crazy picture, and you can wear it at your next Halloween costume. It's so good. I think you know what it is, right? Don't need to explain it. Anyway, thank you.
Hi, everybody. Hi, everybody. We have a couple minutes for questions, but I just have to ask, does kombucha work?
I'm off.
All right,
there's no evidence backing kombucha for your gut or your vagina.
I'm sorry. You just knew it, didn't you?
It's I'm sure someone is going to be looking into it pretty soon because it's pretty popular, isn't it?
Does anybody have any questions? Here we go.
How's it going? Great talk. Just a question about the fecal microbial transplant.
Yep.
Does that have to be preceded by like the germ-free mouse antibiotic? Or will the healthy bacteria outcompete the unhealthy bacteria?
You mean in a human situation? Or in a human situation, it's becoming, I wouldn't say common, but it is becoming an alternative solution for gastroenterologists. So that particular study I showed with the woman that was incredibly sick, that's a very extreme case. But there's a lot of other sort of, cases of colitis or other sort of gastrointestinal diseases that people's health is, or well-being necessarily not crippling, is not okay, and fecal microbial transplant has been offered as an idea. The biggest issue is there's not a lot of good evidence yet supporting how it works well in humans, and I suspect a big part of that is because our individual microbiome is so affected by everything, our genetics and our health and just who we are generally and how we operate. And so that kind of means that on an individual level, there's probably a lot more research that needs to happen before we can fully understand that that's a good answer. That's a good thing that's needed for this particular patient or client. That said, I know that a lot of gastroenterologists are now looking to it and trying it, but I know that there's another issue with finding a good donor. So you want to have someone that's quite close to you, so it's pretty intimate, to share those things. But you definitely want to be comfortable that is not going to make a problem worse and make it better. But I think we're getting there. It's definitely really a big field of interest at the moment. Yeah.
Hi. If someone's had a hysterectomy or gone through menopause, does that change the vaginal microbiome?
Yeah, it does, because what you see, so I can't remember if I mentioned, but I totally meant to, is that there seems to be an association with glycogen, the production of the vaginal mucus and glycogen, with estrogen. It's not completely clear how that relationship works. And estrogen, it cycles over the course of your 28 days in your menstrual cycle, if you're still menstruating, but it changes over the course of your life, so that prepubescent girls often have very low lactobacillus because they have very low estrogen. And then over your reproductive years, up until sort of 45 is the benchmark that we use for perimenopause, that your estrogen starts to drop off to about half of what it was in reproductive years before you hit full menopause, where you have very low estrogen. So something like an estrogen replacement therapy might maybe supplement for that, but it's not completely clear how that link works yet, because the glycogen in perimenopause and menopause still seems to be reduced. Yeah, I don't know if I answered your question fully.
We have a question over here.
Just regarding the graph you showed of the changes with bacterial vaginosis and the fact that we're starting to revert back to the thing after treatment. I don't know a lot about the subject, but I understand that all attempts to change the human microbiome in the gut tend to be short-lived, because it tends to revert to the microbiome that you have when you're about 4 or 5. Is that true, do you think, in the vaginal situation?
I think there is. Yes, for example, in that case, with the woman taking the medications and then you could just see it reverting back, that is a situation that does seem to happen a lot, particularly in that case, which is a very sort of disturbed microbiome. So for whatever reason in that case, I suspect it's something about the behaviour or the genetics or other physiological things that individual is doing, that the treatment works while it's working, but then what we see, particularly in BV, is that almost always there's a remission. after the treatment finishes. It's about 9 months. So you get this response and the lactobacillus dominate, and then there's a remission. Same sort of thing in a fecal microbial transplant, which is sort of like similar but not really, that you could potentially introduce what we would say is the most healthy microbiome into a very sick gastroenterologically bad situation of another individual, but we might not see the result that you would be hoping for. And I think that's where the science is at the moment. We're not so well understanding how the personal medicine is going to be affected. And I think what we're going to see soon is that we'll start to be able to identify specific groups and maybe be able to give, better probiotics or better ways of identifying a healthy situation that could make it more possible.
We have time for one last question here.
Thank you. Great talk. My question is very similar to his, actually. So you mentioned that bacterial vaginosis, if that's how you say it, is affected by what you eat, your lifestyle.
Yeah.
If someone has a unhealthy microbiome, which is not lactobacillus dominant, is there a type of food or diet that they can eat to sort of treat that?
It's a really good question, but there is absolutely... There's very little evidence right now to say that, to say anything with any confidence. So my thinking is yes, there probably is, but we don't know what it is in terms of science.
Okay.
Yeah.
Because my friend Delia, she's like, we're going to go home and eat yogurt. It's like, I don't know if that works like that.
Well, I'll put your yogurt questions. So yogurt is always been a big sort of way to relieve and reintroduce yogurt back into the vaginal tract. And I've heard a lot of women say that they, if they've got BV or they've got irritation, they'll put yogurt on their vagina. But clinical trials have been done and they don't show a response with yogurt. So there's a lactobacillus strain that is used in yogurt, but it's usually a different type of strain. The way sort of bacteria work is that there's genre and then there's species and then there's strains, like there's breeds of a dog. And there can be very, very many of them and they can perform very different functions. And at the moment we're sort of really only starting to understand where they are.
Thank you, Tiffany Nelson, so much.